Hi, my name is Michael Henderson. I am a biochemistry, molecular biology undergrad at Carroll College. The research that is presented today was performed at an RAU, Research Experience for Undergraduates, at Oregon Health and Sciences University. This research was also published in Science Advances journals in November 2020. I'd be happy to provide a link or a copy to anyone who may be interested upon request. So in this study, we explored the use of mRNA therapeutics using mutant epithelial sodium channel subunits. This was achieved by delivery with lipid nanoparticles for treatment of cystic fibrosis. Our research presents data from cell-based models as well as mice with cystic fibrosis. Cystic fibrosis is a devastating autosomal recessive disorder. In a general context, mutations in the CF transmembrane conductance regulator gene results in defects of the CFTR protein. Given that it affects 70,000 patients worldwide, this inability to clear the thick mucosal layer increases chances of bacterial colonization and pulmonary deterioration. The primary channel that is dysregulated is the CFTR channel. This causes ENaC, Epithelial Sodium Channel, to be upregulated. There are over 2,000 different channel mutations in CFTR gene that causes CF. Classification of these group mutations are broken up into 5 classes, and each class affects the protein differently. So in some cases, the protein is not made. In other cases, the protein is not fully functional. Of these 5 classes, the most common CF mutation, delta F508, is a class 2 protein processing mutation. This mutation is caused by a frame shift of a 3 base pair deletion at codon 508 in exon 11 of the CFTR gene. This frame shift causes an absence of a phenylalanine residue. Without this residue, the CFTR protein has a folding defect that causes enhanced degradation of the CFTR channel. But in a majority of studied cases, it is ENaC that is upregulated, causing excessive sodium as well as water reabsorption. So seen here in panel A, in a normal cellular function for lungs, CFTR channel regulates chloride ions, the ENaC channel regulates sodium ions, and aquaporins regulate water flow, altogether functioning and maintaining airway surface liquid, ASL height, for proper mucociliary clearance. Defects seen in a cystic fibrotic lung in panel B for defects of the CFTR protein causes impaired chloride ion transport due to the loss of a functional channel or channel formation. The ENaC channel in CF is not only upregulated, but hyperactivation of ENaC leads to increased water absorption and eventually a formation of a thick mucosal layer that is unable to be cleared from the airways. Therefore, we hypothesized in this study that ENaC activity can be reduced by deploying mRNA that encodes for channel inactivating ENaC alpha mutant with the subunit packaged inside of a lipid nanoparticle for delivery. So ENaC is a heterotrimeric subunit composed of 3 homologous alpha, beta, and gamma subunits, with alpha being essential for a fully functional channel. Therefore, as seen here, our strategy was to mutate ENaC, alpha ENaC subunit mRNA at furin cleavage sites and degenerin sites to alanine, highlighted by the red dots seen here and here. So for proof of successful lipid nanoparticle encapsulations and transfection, dynamic light scattering analysis was performed in HEC293 cells and can be visualized by the cryo-EM imaging on the bottom left panel here. Additionally, we found lipid nanoparticle size distribution was on average 73.52 nanometers with a successful encapsulation efficiency of 99%. For in vitro transfection studies, also in HEC293 cells, images were taken to measure the expression of enhanced green fluorescence protein, GFP, and displayed encapsulation of mRNA with the tag GFP seen in the bottom right panel here. So one of our primary experimental techniques next was electrophysiological assays using the Ussing Chamber. This chamber supports a polarized epithelial cell monolayer. In our experiments, we utilized and polarized CF bronchial epithelial cells on snap well inserts. Upon polarization, these snap well inserts are secured, separating the apical and the basolateral side. Experimenters then are able to manipulate and measure ion transport across the monolayer with various ions, nutrients, or drugs for epithelial tissues. So our CF bronchial epithelial cell lines were derived from actual CF patients and served as in vitro model systems in our electrophysiological assays. In the top left panel seen here, CFBE, cystic fibrosis bronchial epithelial cells, were used and transfected with the LNP containing the mutant ENaC and were tagged with a V5 epitope tag. And with the beta-actin as the gel loading control, we successfully performed a Western blot 72 hours after transfection, showing that ENaC expresses in these cell lines. For our Ussing chamber analysis, we polarized CFBE cells on snap well inserts to allow for tight junction formation. The upper black line seen on these two graphs represent untransfected polarized CFBE cells, while the transfected cells with a lipid nanoparticle encapsulating firefly luciferase mRNA served as our control here. Our experimental, seen on as the red line, represented the CFBE cells transfected with the mutant ENaC via LNP. Upon removal of the snap well and placement into the Ussing chamber, cells were equilibrated with Krebs buffer solution and then treated with amiloride to the apical chamber. Amiloride is a specific ENaC inhibitor, meaning with success in transfection, we'd expect to see less channel formation and overall decrease in amiloride-sensitive currents compared to the controls, which we had seen here and quantified in here. To further confirm our hypothesis, measurements of ASL height, airway surface liquid height, was necessary. To do this, we used transwell inserts this time that aren't removable as the snap wells are, on which for the transwells, the CFBE cells were polarized with ENaC. And with this sheet of epithelia, a semi-permeable membrane was formed across the insert of the chamber. This was done for wild type, untransfected, and alpha mutated here. And with this, after 72 hours of mutant mRNA transfection, we removed the media from the top layer and added cell impermeant rhodamine dextrin, which is red in color. And then the cells were stained with a fluorescent stain called calcium that permeates the cell, which is green in color. This allowed us to distinguish the outline of the cell from the dextrin as it sits on the top of the layer and doesn't permeate the cell. For our analysis, measurements were taken from the top of the cells to where the dextrin had lied on the surface of the membrane. These shapes here on these graphs are representative of the average height of the rhodamine dextrin of 6 individual fixed points of a single transwell insert. The black and gray line representative here of the U mean or the U wild type mean or the alpha mutant mean are mean values of the average single transwell insert. What was seen from the 3D confocal rendered images of the CFBE cells was ASL height had significantly increased in transfected cells. So, given success in vitro, our next step was in vivo studies, with the CFTR knockout mice. For these experiments, nasal potential differences were measured. Nasal potential differences, or NPD, is actually a well-established and powerful technique performed clinically on patients for definitive diagnosis of CF by testing the transepithelial ion transport activities of CFTR and ENaC. Upon oral intubation of mice, tubing was placed in the nasal respiratory epithelia to measure the voltage across the nasal epithelium. Ringer's buffer, containing amiloride that inhibits ENaC, is then injected after the steady state of the mice with the normal Ringer's buffer. With the NPD setup only being possible in a few labs around the world, we present promising data of a decrease in amiloride-sensitive current seen in the comparison between the wild-type mice and CFTR knockout mice, with the gray line representing the baseline reading, red line representing the transfected mice, and the blue representing the extinction of the reduced reading. So, in summary, dysregulation of ENaC is common across many CFTR gene mutations. α-ENaC subunit mRNA, as we have presented here, can successfully be encapsulated within lipid nanoparticles. And furthermore, alpha-mutant ENaC transfection not only reduces amiloride-sensitive currents in ENaC in vitro and in vivo, but it increases airway surface liquid height in airway cells. For our conclusion, our results from this research study showcase the promise of mRNA therapeutics as a potential universal treatment for cystic fibrosis, regardless of underlying CFTR mutation. And seen here in the intervention, our goal is to ultimately restore airway surface liquid height to allow for proper mucociliary clearance, preventing the formation of the thick mucosal layer that often leads to pulmonary deterioration. As acknowledgements, I would like to first acknowledge the partnership between Oregon State University and Oregon Health and Sciences University, with the Sahai Lab specializing in lipid nanoparticle formulation, and the McDonald Lab specializing in CF treatment. I would also like to acknowledge my direct mentor, Nanda Mukherjee, for his advice and his mentorship and his skill set and ability to assist me on these and teach me these procedures on these projects. And I would also like to acknowledge that this opportunity would not have been possible without the faculty and staff support of Carroll College in seeking out this opportunity. And I would also like to thank our funders of the National Heart, Lung, and Blood Institute and the Cystic Fibrosis Foundation for funding. And with that, thank you so much for attending the presentation today. And I'd be happy to connect with any of you who may be interested in learning more about cystic fibrosis or how gene therapeutics may be the path for the future in ultimately treating and eradicating rare diseases.